diseases. And so in a previous chapter, we talked about monogenic inheritance and genes that are either a single gene that is like a single cistron with a single promoter and then a single RNA transcript, or they at least behave like they're a single gene. And that is, they may be a complex of genes that the transcripts all get spliced together before they're translated. That's how you can have something that's multiple genes but still behaves like a single gene. Chapter 9 is all about complex diseases and the issues with these complex diseases. All right? So some key terms from chapter 9. First one, endemic or non-communicable diseases. These are two terms for the same thing. And they're basically diseases that have an incredibly heavy genetic component, and therefore most of the pathology is the result of your genetic programming. Okay? Most, if not all, of the pathology is associated with your genetic programming. So you can call them endemic diseases or non-communicable diseases. So they're diseases that you cannot pass horizontally. You cannot pass them from individual to individual except vertically by passing them on to your offspring. Okay? These are genetic illnesses with a heavy degree of heritability. Okay? Now, anyways, sorry. They're still not monogenic diseases if it's a complex disease. So we have this next term. It's called familial aggregation. And so familial aggregation is this is how you would define this term. It's saying that these non-communicable diseases are more common in certain families, but they do not adhere to normal Mendelian inheritance patterns. Okay? So they're more common in certain families because of the genetic programming, but they do not adhere to normal Mendelian ratios. Okay? They are not monogenic diseases. These are complex diseases that are either oligogenic, meaning few genes, or polygenic, meaning many genes or multiple genes. Okay? So these definitely run in families still, but they're much more complex than, say, cystic fibrosis. That's a monogenic disease. Okay, where the inheritance pattern is going to be very predictable. These still run in families, but it's not as predictable that if your mom and your dad have that genetic illness, then you're guaranteed to have it. Okay?
Can you give an example of one?
Asthma, type 2 and type 1 diabetes, Alzheimer's, most of our cardiovascular diseases. Several types of cancers are going to be this way as well. All right, this one's fun. This one came up in the chapter, and it was just kind of like, hey, this is a fun idea applied to genetics. The first law of technology. The first law of technology says we overestimate short-term impact and underestimate long-term impact of a new technology. First law of technology, we overestimate the short-term impact and underestimate the long-term impact of a particular technology. And the way this is applied to genomics is that, I mean, you probably heard things like this. I know you guys are all young enough that you were born either the year that all of these publications came out, or even shortly thereafter. Most of the publications as a result of the Human Genome Project came out in 1999. So some of you may be old enough that you were born slightly before that, but you're all born sometime around there. But you still hear comments like this, that knowing the human genome is going to revolutionize medicine. It's going to individualize medicine, right? So we can treat people specific to that person because of their genetic programming. Okay? And this is all a result of the first law of technology. We're going to overestimate the short-term impact of it. But we're also going to underestimate the long-term impact because they're just things that we don't anticipate being a result. Like the epigenetic revolution that came as a result of mapping the Human Genome Project. Okay? That was something nobody could have, well, not nobody, but very few people hypothesized or predicted because everybody thought it was going to be super simple. you were going to find a gene that was going to basically explain all human behavior and all human conditions. All right. Heterogeneous. So heterogeneous means that the same phenotype is the result of several different genotypes. Okay? That the same phenotype or the same character would be the same trait. It's the same trait for a particular character can result from several different genotypes. And where it gets really complicated and would still fall under this category of heterogeneous is that the genotype at several different loci can result in the same trait for a particular character. And a good example of this that would not be a genetic illness would be eye color. Okay? There are at least 15 different genes involved in determining human eye color. And so there are many different ways in which you can end up being a person that deposits very little pigment in your iris and get something that's gets something like a blue eye color, okay? And several different genetic loci that various genotypes there could end up resulting in the same phenotype. All right? Heterogeneous. Next one, lambda R. This is fun. This is one way in which you can quantify the heritability of a complex genetic character. Okay? This is one way in which you can quantify the heritability of a complex genetic character. That's all I'm going to say to define it, and then we'll talk about it in more detail in a moment. I've got a whole slide. You're going to love it. You're just going to learn so much from it. You're going to walk out of this class feeling fulfilled, warm. Yeah, that's pretty much it. I don't know how else you'll feel, but however else you feel is not my fault. It's someone else's fault. Or your own. The next one is heritability. And so heritability has basically two meanings. One is just a general meaning of heritability, where it's basically like, how easy is it for you to pass on a trait, right? How easy is it for you to pass on a particular trait? Like red hair, for instance. And I don't know if you noticed this or not, but I am a redhead. Neither of these are redheads. And our oldest, she's not a redhead either. And it's because red hair has a low heritability. Okay? But heritability also has a very specific meaning, and it is another way to quantify the heritability of a trait. And you're like, how can heritability be a specific way to quantify heritability? And that's because, again, it has two different meanings. It has a general meaning, just how easy is it to pass on a trait, but then it has a very specific meaning, which is a mathematical formula. Again, I'll show you this in a little bit. It's not time yet. It's not time yet. In a little bit, it will be time. Next term, quantitative traits, or QTs. These are traits that vary across the spectrum. Where basically you've got a minimum value and a maximum value, and anything in between is possible. Okay? Quantitative traits. Best example of a quantitative trait is height. Human height. You've got a minimum value, a maximum value, everything in between is possible. And you're like, what is the minimum value possible? I don't know. You'd have to ask a biophysicist or a historian, right? Although you're like, well, could it be possible that somebody could be shorter than the shortest person who's ever lived? Maybe. You'd have to talk to a biophysicist and talk about, biomechanical necessity. But anyways, with human height, you've got a minimum value, a maximum value, anything in between is possible. Okay? This would be compared to like a discrete trait. Like hitchhiker's thumb. It's yes or no. Right? Yes or no. But you're like, wait, that's impressive, Dr. Eagle. That's a little bit more than just a hitchhiker's thumb. You're right, but it's a discrete trait. It's like nobody talks about this and how impressive. I mean, that's 90 degrees. But it's, it's a bit anachronistic because people don't really pick up hitchhikers anymore. So.
You get your PhD?
I think so. Yeah, I think so. So, I mean, it had to. Yeah. Next term and the final term. Genome-wide association studies. Genome-wide association studies, or GWAS. And these we're going to talk a lot more about in chapter 10, which won't happen until next week. We'll talk a lot more about GWAs in chapter 10, but right now, this is where you try to find the genes associated with particular characters. You search the entire genome to find all of the genes associated with a particular character, like human height, or human eye color, or the ability to build muscle mass, or a particular genetic illness. Okay, genome-wide association studies. Again, you're hunting for every gene that is involved in a particular character. Yeah. He wants to get up here? Okay. So, first question, what do we know about complex diseases? What do we know about complex diseases? All right, we actually know quite a bit about them. First thing is the frequency of these diseases is the frequency of a particular complex disease is typically much greater than the monogenic diseases. Monogenic diseases, I don't know if you remember when we were talking through these, but they tend to be incredibly rare. Now, there are a few exceptions, but they tend to be very rare. Complex diseases tend to be fairly common. Like, for instance, some of our cardiovascular diseases that have a high heritability they have prevalence like 1 in 10 or 1 in 5. Okay? I mean, some of these are really, really high. And then if you complex all cardiovascular diseases together as a single complex genetic illness, now you've got something that's like 1 in 3, the prevalence. I mean, these are really, really high. They show familial aggregation. That is, they are more common in certain families, but they do not conform to Mendelian frequencies. So you can't talk about things as dominant and recessive, right? Those terms mean nothing for these complex genetic illnesses. Which is nice. Because then it's not, well, one, it's nice because then you don't have to do like, chi-squared analyses to figure out if your, crop of offspring are matching what the predicted values are. But it's also nice because it forces you to anticipate that environment's going to have a really large role in these diseases. These, unlike monogenic genetic illnesses, these are more frequent in post-reproductive age. Okay? Complex genetic diseases are more prevalent in people that are post-reproductive age, unlike monogenic genetic illnesses. Really, the only monogenic genetic illness that is really common in post-reproductive age is Huntington's disease. And that's because if it wasn't, because it's fatal, that it would natural selection would eliminate it. Okay? And that's kind of the idea of like, well, some of these genetic illnesses that are the result of something that shows up is the only reason they still persist is because it happens before they reach sexual maturity, and they pass on, and then that individual with that genetic illness, either they survive to reproduce or they don't, and it's just, carriers keep it going, right? People that are carriers, or for those that are dominant inheritance, you're talking about different levels of severity, right? So if it's a dominantly inherited monogenic genetic illness, if you're homozygous dominant, you usually have a very advanced form of the genetic illness and don't reproduce. And if you're heterozygous, you still have the genetic illness, but a less advanced form, and you can still reproduce and pass that on. Another thing we know about this is the prevalence for many of these complex diseases is increasing. Every generation, the prevalence of these genetic illnesses increase. And the question is why? And there's really two possibilities. One is we are changing genetically, right? That every generation we are changing genetically, which we know is true. Or two, that every generation the environment that we are exposed to is changing, which we also know is true. And so we're like, oh, it's probably a combination of the two. But anyways, we'll get there in the latter part of chapter 9 on Wednesday. And then, second to last thing we know about these complex diseases is that they're often comorbid with other diseases. And what that means is where you have multiple pathologies appearing simultaneously. So a lot of these complex genetic illnesses are comorbid. And then the last one, and this is one that you should be able to predict with complex genetic illnesses because you've got multiple genes involved. Environment is going to heavily influence the expression of the program. And this makes sense even outside of diseases, but just complex genetics characters, like human height. Human height is obviously one where environment's going to play a huge role, right? A lot of it's genetic, but a lot of it's environment as well. Yeah. Okay. Any questions about this question? That's one of my favorite questions to ask. Yeah, Alyssa.
So, not about this question, but about one of the common terms, the endemic non-communicable. What's the difference between saying that and a complex disease?
In a complex it's just a matter of perspective. So when you're talking about endemic non-communicable diseases, you're looking at it at the perspective of the population. Right? Endemic, it's isolated to a particular individual with a particular genetic programming, and it can't spread through a population except vertically. And then when you're talking about complex genetic illness, your perspective is basically the genome, the individual genome.
But they mean kind of the same thing.
Correct. Okay. Yep. They sure do. Do you want me to go back and write endemic, comma, non-communicable, comma, complex? Okay. Well, because even monogenic genetic illnesses are still endemic, non-communicable diseases, but they're not complex genetic illnesses. Yeah.
Do they know about these because of genome-wide association studies? Is that how they know about the cardiovascular diseases being passed down in this?
Well, you can do heritability studies without any information from the genome. We've been doing these for decades without any information from the genome and the sequences. That sure helps, but you can do heritability studies without that. And because I'll show you the two ways in which we quantify heritability, and you don't need to know anything from genomics. But as far as, it obviously does have some applications. And you'll see that as well. All right, what are the challenges with treating complex diseases? What are the challenges with treating complex diseases? First challenge is individuals are too heterogeneous, meaning you and I might have the same complex genetic illness. For instance, you and I may both develop a certain type of cardiovascular disease, with a high prevalence, say 1 in 5 or 1 in 8. Okay, you and I may develop that same cardiovascular disease, but have different genetic programming that made us, both susceptible to it. Or you and I may both develop type 2 diabetes, but we have two very different genetic susceptibilities to it. Okay? And that's what we mean by heterogeneous. So it makes it really difficult. It's not as simple as monogenic, where you've got a single locus, and it's like if you've got that genetic illness, you can predict they're going to have a specific mutation at that locus. It doesn't work that way for complex genetic illnesses. Other one is even if somebody knows that they have an increased risk for a particular genetic illness, they oftentimes won't seek preventative care. It's the same thing like when you tell somebody, hey, smoking cigarettes is a bad idea because it correlates very significantly with developing lung cancer. They just don't care. It's like, I want to smoke cigarettes. I like the way it tastes. I like the way it turns my fingertips brown and my teeth brown. I just love it. Can't get enough of it. Sorry, that was kind of pejorative, but you know, what are you gonna do? Yeah.
What do you mean by preventative care?
Preventative care, because with these complex genetic illnesses, we can shift medical treatment. We can't really, we're not anywhere close to being able to individualize treatment, where it's like I can look at your genome and I know exactly how I need to treat asthma in you versus in me. But what we can do is we can shift from diagnose and treat to predict and prevent, right? So when we know, like if you submit a DNA sample and we look just at single nucleotide polymorphisms and your genetic propensity for developing asthma or developing certain types of cancer, to where you may have an increased likelihood. And so now I can shift medicine from just waiting until you're diagnosed with that cancer and treating it, to now I can say you've got you're 20 times more likely to develop this. So let's go through these preventative measures, right? Let's increase your antioxidant uptake so you can decrease the rate of mutations, right? In your cell lines. Right? Or let's increase the frequency of your screening. So even if you do develop cancer, we can catch it earlier. Right? Or if it's a cancer that develops in a tissue that you don't really need, some of that preventative treatment might be removing that tissue. Right? And so that's what we mean here is with these complex genetic illnesses, even if you know you have an increased likelihood for developing some kind of cardiovascular disease, oftentimes what you find is people don't care, right? Like let's say you're 5 times more likely to develop or to have some kind of cardiovascular disease and that results in congestive heart failure. And so we're like, okay, so we know this, so let's really focus on preventative care, and you should really avoid diets that are rich in these types of foods, which when combined with your genetic propensity are going to increase plaque buildup in your arteries and are going to make it significantly more likely that you're going to develop this by a certain age. Okay? So we can do that. We can do that because we can know if you have an increased likelihood to developing one of these kinds of diseases. The thing is, people don't care, right? Or I can tell you, like, you're 10 times more likely to develop type 2 diabetes. And so we're like, you should probably develop, by the time you're late 20s, early 30s, you should develop a regular exercise routine and stick to, you know, this kind of diet. And then you probably won't ever develop it. And it's like, people don't care. It's like, no, I want to eat pizza and donuts every day, and I don't want to exercise. I totally get it. Listen, I'm not saying like this is some other people. I love pizza and donuts, and I hate exercising. So that's what I mean by here. Does that make sense? But doing this, even though it's really difficult to treat these genetic illnesses with what we know about the human genome, we can shift medicine to predict and prevent. Right? We can shift that. Or if you have an increased likelihood for developing Alzheimer's, there are things that you can do, brain exercises, that puts off the development of the dementia. So, all right. Another thing is the relationship between nature and nurture is very complex. So with these complex genetic illnesses, all of the things involved in determining whether or not a specific genotype is going to be expressed, it's super complicated. Super complicated. And we still don't understand a lot of these. We're just getting started in the area of epigenetics, which is basically understanding how your environment shapes the expression of genes. Right? We're just getting started. It's in its infancy. And yet, it's probably more important than your programming. Right? Your epigenome is probably more important for predicting these genetic illnesses than your actual genome. And it's like, wow, it's weird. And we don't know very much about it at all. And we still don't know very much about the genome, even though we haven't mapped most of it. About 96.5% of it mapped, but we still don't understand it very well. All right. Next question, is it nature or is it nurture? And the answer is, of course, it's both, right? It's both nature and nurture, especially with these complex genetic illnesses, because we just said that the environment plays a very large role in determining phenotype. So it's obviously going to be both, but the question is, how do we quantify what percentage of it is programming? Okay? And there are two ways that we can do that. One is using lambda R. And so in this, we divide the familial aggregation by the population frequency. And so what that looks like is this. Lambda R is equal to actually, hang on. Don't write that yet. Don't write that yet. Madison, I need this. Thank you. Put prevalence on the bottom of this. And on the numerator, I'm gonna structure it this way. The percentage of identical twins both displaying the trait. The percentage of identical twins both displaying whatever that trait is. Okay? And that way we're keeping it general. It doesn't have to be a genetic illness. That trait could be a genetic illness. And so lambda R is equal to 1 when genetics does nothing. So this lambda R would equal 1 when basically it's all environmental, right? So this would be things that are like pathogen, how do I want to say this? Pathogen-induced diseases. For most, lambda R is large for monogenic conditions. And this should make sense because if it's monogenic and it's a single gene predicting this phenotype, your percentage of identical twins displaying that trait should be really, really high, like close to 100%. So for example, lambda R for something like cystic fibrosis is like 500. Okay, and that would be something that's really large. Okay?
How can it be over 100?
How can it be over 100? Because the prevalence in the population is almost always less than 1. For this to be 1, 100% of the people in the population would have to have that genetic illness. Right? Because we're dividing it by prevalence. Prevalence is a percentage of the population with the particular genetic condition. Yeah?
Is there like a threshold that you have to reach for it to be considered monogenic?
No, it's just monogenic conditions are going to tend to have really high lambda R values. You wouldn't use this to predict whether or not it's monogenic or not. You would use a pedigree. So if you do a genetic pedigree for a family and it's conforming to Mendelian frequencies, then it's monogenic, right? You would do that because it'd be a lot simpler. Okay? And so lambda R, if it's greater than 1, if lambda R is greater than 1, there's some genetic component. And for these complex genetic illnesses, like asthma, type 2 and type 1 diabetes, several different types of cardiovascular disease, the lambda R values are somewhere in between like 10 and 20 for a lot of these. So where there's some genetic component, but still a lot of it's environmental. All right. The higher the prevalence, the lower the value. This should make sense, and this is why complex genetic illnesses tend to have lower lambda R values than monogenic conditions, because complex genetic illnesses are more prevalent, right? That was something we talked about at the beginning. They're more prevalent than monogenic genetic illnesses, and so when you're dividing by a larger denominator, your fraction gets smaller, right? And so as your prevalence increases, your lambda R gets smaller. Monogenic diseases have high values. We already mentioned that. Okay, so some complications here. That should say relatives, not relative. Relatives. So the complication with lambda R is that relatives have a very similar environment typically, especially identical twins. Identical twins don't always share a placenta, but they often do. And so they're sharing a placenta, a womb, a mom, right? And so it's like, man, that is most likely the most important environmental factor in all of your development, is what you experience in utero. And so you're like, oh yeah, but can't we do these studies on identical twins that were put up for adoption and so grew up in different homes? You can, but the most important part of their environment in determining their development happens in utero. And so it's like they've already shared the most consistent part of their environment. So you can, but then it's also complicated. There are ethical concerns, involved in this, because a lot of these adoptions are closed. If you're going to put identical twins up for adoption, they get adopted by different homes. So anyways, it's complicated. So lambda R is useful, but it's difficult because it relies so heavily on comparing it to the overall prevalence. Okay? Heritability, this is where heritability is sometimes nicer than lambda R. So in heritability, may I erase this? We all have this written down? Okay. Heritability, which we represent as h squared, and that's just to demonstrate that we're talking about a very specific form of heritability. It's not because we're squaring some value. Do you guys want to leave?
Yeah.
Okay. There's stuff in my office. I'll just bring it home with me.
It's open.
Oh, okay, then you can grab it.
Sorry.
And so for heritability, we divide basically this value called VG by total variation. Okay, and so what VG is, it's the proportion of the variation that's due to genetic differences. And then V is the total variation. And so these are values that we really don't care how prevalent the illness is. The prevalence of the illness in the population it really doesn't matter. We are concerned with total variation, which the prevalence is going to influence total variation. But really, we're just concerned with what percentage of this is due to genetics. Are you going to ask how we determine that?
No, I was going to
Okay.
say, why can't you just cancel out V?
Because these are different variables. So VG, VG is not the same thing as V times G.
Okay.
Yeah, this isn't V times G, this is V of G. Yeah. That's a good question. Why can't we just cancel out V? Because it's not the same V.
Okay. So VG,
Here, let's do this.
like maybe little g.
Look at that. I mean, let's just do G in parentheses. Let's just do whatever we want. I mean, most of the time we write V and then in parentheses G, but heck, I mean, we write whatever we want. It's our class. We do what we want. So the way we determine this is we do a correlational study. We basically, we study a population. We study a population. We put the phenotype on our y-axis, the various frequencies of all of the different genotypes on the x-axis. Okay? So again, the phenotypic conditions on the y-axis. And you're like, how do we do that for some things where it's like yes or no? You don't do a linear regression, right? You can't do a linear regression if your dependent variable is discrete. We all know that, right? You can do a logistic regression. In order to do a linear regression, your dependent variable has to be continuous. Right? Yeah, you all know that. Like human height, these quantitative traits. Human height, that's an excellent dependent variable, right? So let's take human height and let's put it on the y-axis. Human height. And then, you know, because the US system is terrible, let's do human height in meters. You're like, no, Dr. Neal, this is America. I will not use meters. That's your loss. Way better. So human height in meters, on the x-axis, we're going to put all the different genotypes. Genotype frequencies. And what we're looking at is just from the different genetics, how much of the variation in this can we predict? Right? How much of the variation in human height can we predict just from differences in the genotype? Nothing else. We're not putting in any information about their environmental factors. We don't care where they grew up. I don't care what their parents fed them. I don't care if they were breastfed or bottle fed. If they were given goat's milk or cow's milk, or God forbid, soy milk. I don't care, because all I'm putting on the x-axis, my only independent variable is their different program. Okay? We're just putting in the different programs. And then we're going to see how much of this variation can we predict. And so we actually don't need to know really anything about the genome, because we were doing this when all we knew about were single nucleotide polymorphisms. And we just put all the different combinations there, and then we'd figure out how much of the variation in human height we could predict from just genetic differences. And for some of these, like human height has a really high heritability. It's like 80%. 80% of the variation in human height is predicted by genetic differences. Nathan, did you have something?
Yeah. How do we know that the particular sample we chose to test didn't happen to have height that had to do with their diet and habits to correlate?
I mean, you just do multiple. I mean, you do multiple samples. You wouldn't try to set a generalization for the entire human population from a sample of Eastern Europeans, right? I mean, you'd have to sample a very broad collection of the human population, which we've been doing. I mean, the beautiful thing is a developed country is a wonderful place to sample the entire world's population, other than the incredibly, extremely isolated groups. And you're like, they're going to be wonky genetically anyways, because they're reproductively isolated from the rest of humans, right? And so it's like, do we really care about those data? Not really for this, because we know that they're going to be unusual genetically, because they're reproductively isolated, right? But if you just sampled, if you're just like, you put up a sign and you're like, free tacos to everybody that gives a spit sample, right? And you set it up like in downtown LA, man, you're going to get a pretty good mix of people who are going to show up. I'll give you my saliva for some free tacos, right? You're going to get people from all walks of life. Rich people like free tacos. Poor people like free tacos, right? And LA has one of the most diverse populations found anywhere on Earth. I mean, yeah, and it's not very far away, you know? It's very convenient. John, did you have a question?
Yeah, well, I thought you were saying that human height was more based on environment.
No, it still has a heavy environmental component. So, like a monogenic condition, environment plays no role. Okay. Virtually no role. So it's like, its heritability is 100%. And so even human height, you're like 80%, that's a lot. But that still means 20% of the variation in human height is environmental. That's that's a very significant proportion.
I thought you meant like 80% is environmental.
Oh no, if human height, 80% is genetic, 20% is environmental. Which, now for human height, that's a lot. I'll show you oh my gosh, I won't show you a slide today because we're out of time. I'll show you a slide on Wednesday where we'll actually go through some of the, I mean, you have the slides now, they're on Canvas, you can go look, take a little peek, but without the explanation. Have a wonderful day. Enjoy the rest of your Monday.
Do we have a quiz before Wednesday?
No.